Hello, the topic we are discussing today is genetic and environmental influences on prenatal development. The objectives of this module are: at the end of this chapter, the student will be able to understand the genetic influence on development. They will know how cell division occurs, and they will be familiar with different concepts like genotype and phenotype. They will understand the interaction of genetic and environmental factors on development. The life of human being develops from a single cell called zygote, which is created when a male reproductive cell called sperm unites with the ovum, the female reproductive cell. These male and female reproductive cells are known as gametes, each containing huge amounts of genetic information. The child is influenced by a vast array of genetic and environmental factors. The transactions between genes and environment make each child unique. The concepts of genotype and phenotype provide a framework about the interactions of genes and environment. Genotype is the particular set of genes a person inherits from its parents, for example, height and eye color. Phenotype is the visible expression of the person's physical and behavioral characteristics created by the interaction of a person's genotype with the environment, example, motor abilities and intellectual skills. The process of genetic transformation occurs in the following manner. The egg or ovum, which is the largest cell, which is 90,000 times heavier than sperm, slowly travels through the fallopian tube, and if sperm are present at that location, one may unite with the ovum in a process called fertilization. In the process of fertilization, only one sperm can enter the egg's outer membrane. The moment the sperm penetrates the egg is called conception, creating a new and separate cell called the zygote. Chromosomes and genes are located inside a cellular structure called nucleus. During conception, 23 chromosomes from sperm and 23 chromosomes from egg join to create 23 chromosome pairs. Each chromosome contributed by the father's sperm is homologous, that is, similar in shape and function, to one of the chromosomes contributed by the mother's egg. Copies of these original 23 homologous pairs of chromosomes are passed on to every cell except reproductive cells, 23 chromosomes. During its development, that is, the zygote's development, it undergoes a special form of cell division called meiosis, in which 23 chromosome pairs are halved. Now, 23 chromosomes from sperm combine with 23 chromosomes from the egg to produce the correct number of 46 chromosomes for a new human being. Genetic variability is added during meiosis by a process called crossing over, in which equivalent sections of homologous chromosomes randomly switch places so that genetic information is shuffled even more. Let us look at this figure to understand the process of meiosis. In A, we see the results of this replication. In B, crossing over between chromosomes ensures the zygote's unique genetic inheritance. In C, the male chromosome pairs separate to form two cells, each with 23 chromosomes. In the female, two cells are also formed, but one is non-functional and may or may not produce two more non-functional cells. In D, the chromosome separates once again, in the male forming 4 sperm cells and in the female a single ovum and a fourth non-functional cell. In E, we see a zygote is formed, and finally the F figure shows 23 chromosome pairs. Mitosis occurs in both autosomes, that is, chromosomes that contain matching pairs, and also in sex chromosomes. A cell duplicates its chromosomes and then divides into daughter cells that have the exact same number of chromosomes as their parent cell. Thus, zygote divides and continues to divide, each time producing new cells that have the full complement of 46 chromosomes and gradually becoming a multicellular organism. In this figure, in A, we see a zygote with only 4 chromosomes rather than the 46. In B, we see each chromosome splits in half lengthwise to produce a duplicate of itself. In C, the duplicates move away from each other as the cells begin to divide. In D, the cell has divided in two, and each new cell has the same set of chromosomes as the other and as the original parent cell. Chromosomes are bound by molecules of deoxyribonucleic acid or DNA, which are made up of nucleotide. These store genetic information and transmit it during reproduction. Genes are the portions of DNA molecule are located at particular sites on the chromosome where they code for the production of certain kinds of protein. Each of the many different kinds of proteins in the human body serves a different function. With respect to behavior, genes never work in isolation, but always in combination with environmental influences. Let us look at how the transmission of traits takes place. There are two basic concepts that play a crucial role in understanding influences on development. First, at any given gene's position on two chromosomes, there can be alternate forms of that gene called alleles. For example, if there is gene A, it can have two alleles, the dominant allele represented by capital A and the recessive allele represented by small a. Each of these alleles are inherited from either of the parent. Second, if the alleles inherited from both the parents are identical, that is, either two dominant alleles or recessive alleles, the person is said to be homozygous. If the alleles are different, that is, one dominant and other recessive, the person is heterozygous. If capital A represents the dominant allele and small a represents the recessive allele, the individual can have one of three possible combinations, that is, homozygous dominant represented by two capital A's or homozygous recessive represented by small a's or heterozygous represented by the letters capital A and small a. When there is a combination of heterozygous alleles, the result may vary. First possibility is that the combination of two dissimilar alleles will produce an outcome intermediate between the traits for which each single allele codes, that is, light-skinned plus dark-skinned gives rise to intermediate color. This is called incomplete dominance. Second possibility is that both alleles will express their traits simultaneously. For example, the allele for blood type A in combination with the allele for blood type B produces the blood type AB, which has both kind of antigens. This is called codominance of the two alleles. The third possibility is that the character associated with only one of these alleles may be expressed. Only the character encoded by the dominant allele is expressed over the recessive allele. This is called complete dominance. Humans have 23 pairs of chromosomes, out of which 22 pairs are autosomes and one pair is the sex chromosome. A female has two X chromosomes, one from mother and another from father. A male has one X chromosome from his mother and a Y chromosome from father. The pattern is referred to as XY. X chromosome is 5 times longer than a Y chromosome, therefore it carries more genes. In males, some genes on the X chromosome have no equivalent genes on the Y chromosome. As the male inherits one X chromosome, that is X-linked genes from the mother, and if this X chromosome carries a harmful recessive allele, the associated genetic disorder will automatically be expressed. Hemophilia is an example of X-linked recessive trait. Most characters like intelligence, creativity, sociability, style of emotional expression are all influenced by interaction of multiple genes. Development of traits depends on configuration of genes and is not likely to be passed on from parent to child. For example, individuals who are born geniuses are sometimes born to parents of quite ordinary intelligence, and geniuses sometimes produce children who are not talented. Let us look at few of the genetic disorders. Genes can have both positive and negative effect. Then how do harmful alleles survive? A harmful allele is not harmful in the heterozygous state. They are beneficial in combination with a normal allele. For example, Phenylketonuria or PKU is a disease caused by a recessive allele that fails to produce an enzyme necessary to metabolize the protein which is present in milk. In this figure, when both parents carry recessive allele for PKU, they have 1 in 4 chance of producing a child with a disorder. If their dominant genes for normalcy are passed to their offspring, the child will be normal. If the child receives one dominant and one recessive gene, it will be a carrier. The child will not have the disorder then. If it receives recessive genes from both the parents, it will have PKU. Now let us look at few of the chromosomal abnormalities. Developmental disorders are not only caused by genes or gene groups, but by defects in entire chromosomes. Chromosome defects are not present in a child's parents. Instead, they are generally arise during the process of meiosis when eggs or sperms are formed. That is, sometimes when a gamete is formed, the male's sperm and female's ovum do not have their normal set of 23 chromosomes. The most notable example of chromosomal abnormalities is Down syndrome and abnormalities of the sex chromosomes. For example, Down syndrome or trisomy 21 is caused due to the presence of an extra full or partial chromosome. An individual with Down syndrome has a round face, flattened skull, an extra fold of skin over the eyelids, a protruding tongue, has short limbs and retardation of motor and mental abilities. The syndrome makes the individual susceptible towards leukemia, heart disorders and respiratory infections. Now a newborn baby normally has an X and a Y chromosome or has two X chromosomes. Human embryo must possess at least one X chromosome to be viable. The most common sex-linked chromosomal abnormalities involve the presence of an extra chromosome, either an X or Y, or the absence of one X chromosome in females. Turner syndrome is a chromosomal disorder in females in which either an X chromosome is missing, making the person XO instead of XX, or the second X chromosome is partially deleted. These females are short in stature and have a webbed neck. They might be infertile and have difficulty in understanding mathematics, but their verbal ability is often quite good. In Klinefelter syndrome, a male inherits extra X chromosome. There are some odd physical characteristics such as undeveloped testes, enlarged breasts, and are tall. They also suffer from sterility and mild to severe cognitive difficulties. concept of the range of reaction explains that heredity does not rigidly fix behavior, but instead establishes a range of possible developmental outcomes that may occur in response to different environments. When a reaction is extremely narrow, it is said to exhibit canalization. With a highly canalized state, there are few pathways that development can take. Therefore, intense or more specific environmental pushes are required to deflect the course of development. Gilbert Gottlieb provides a modified view of gene-environment interaction. According to them, genes play a less determinative role in shaping development. Individual development is organized into multiple levels, that is, genetic activity, neural activity, behavior and environment, all of which influence each other. Let us look at the bidirectional influence in gene-environment interactions. In this figure, from bottom to top and from top to bottom, genes and environment mutually influence each other. Let us see how the effect of genetic makeup helps in shaping the environment. We all know that environment influences genetic expression, but the notion that environment can be shaped by genes is relatively new. According to a study conducted in the year 2000 by Hetherington and Plowman, people's genetic makeup can influence their environment. There are three ways in how this occurs. The three ways in which people's genetic makeup can influence and shape their environments is passive, evocative and active. Firstly, let us look at the passive genetic environmental interaction. The interaction of the environment created by parents with particular genetic predispositions which encourage the expression of these tendencies in their children. Secondly, the evocative genetic environmental interaction. Individuals' inherited tendencies may evoke certain responses from others, as child's inborn tendency to smile may elicit smiles from others. Thirdly, the active genetic environmental interaction says each person's genetic makeup may encourage him to seek out experiences compatible with their inherited tendencies. In a process called niche picking, people search for, select or build environments compatible with their predispositions. If individuals are gregarious, they tend to seek out social activities. According to human behavior genetics, there is a significant difference in individual development due to heritability factors. Studies have been conducted on family members with known degrees of biological relatedness, such as monozygotic and dizygotic twins and adopted children, as well as the degree to which they inhabit shared or non-shared environments. It has been observed that individual differences in intelligence, spatial skills and verbal proficiencies are influenced by genetic factors, while temperament, personality, emotionality are also influenced by genes, but the influence declines with age, as people's personalities are increasingly influenced by life experiences. At last, it may be concluded that a child's personality in later life is dependent on a considerable degree of the goodness of fit between the child's temperament and the child's rearing environment.